So here in this picture, it's basically, kind of like sea anemone here. The cell is dividing, it's ripping, and whatever, so for each of the daughter cells, so you have a parent cell, and once it divides, you have daughter cells. These daughter cells are going to have the identical DNA because it's asexual reproduction happening here. So the DNA, the cells must first replicate its genome. So it copies its DNA and then it divides. But in the end, both of these cells are going to have the same exact genome or DNA. So same exact genetic information. Sometimes what happens is that the cells, when they are dividing, something goes wrong and they have maybe one extra copy. And one disease that you might have heard about is Down syndrome. So that's trisomy, I think 22 or 23. There's one extra copy of one chromosome, and then that person who has that mutation, you can call it, or extra copy, then they have the Down syndrome. So when they divide, one copy remains in this cell and the other copy remains in this cell. But if there are some problems during the division, then it can cause some diseases. Okay, so next process we're going to talk about is DNA replication. So before I get started on this, I want to know if there are any questions on the content that we have covered so far. Yes, no? No, okay. Okay, so DNA replication, we're going to cover the steps for it. You need to know the enzymes. There is one question on the quiz that's going to ask you the DNA replication enzymes. So make sure you know the enzymes and their function. So DNA replication occurs in 3 major steps. Think of this. So you have the chromosome, looks like letter X here. That's what I showed you before in the nucleus. Zoom in on it. You see the double helix structure of DNA. It has the genetic information, and this genetic information needs to be read in order to make the protein. So think of this as a book, as a closed book. And in order to read the information that's inside, this book needs to be open. So first enzyme that comes in is helicase. So this is the name of the enzyme. This helicase is going to open this double-stranded DNA or double helix DNA. And if you guys remember, these bases, they're linked through hydrogen bonds, which are weak bonds. So this enzyme comes in, it just unzips this DNA. Just unzips this DNA. Once that happens, another enzyme called DNA polymerase, please do not confuse this with RNA polymerase in transcription. We're talking about a different process, that's replication, making a copy of the DNA before you do the transcription. So DNA polymerase, it's going to come and it's going to attach wherever, it's going to start the replication, and then it's going to start grabbing these nucleotides. These nucleotides just float around in the nucleus, and it's going to start adding the complementary base pair. So let's start here. So if it sees a C, it's going to grab a G and add that. Another C, it grabs another G. Then next one, it reads G, then it grabs another C. So it's going to create a complementary strand. So complementary means G is always going to go with C, C is always going to go with G, A is going to go with T. So it does that. That's the job of DNA polymerase. So if I ask you on the quiz, what is the function of DNA polymerase? You're going to say it's going to assemble. It's going to assemble the new strand of DNA. And in addition, it also proofreads to make sure there are no more mistakes, because this is happening so much in your body. It wants to make sure there are no mistakes, but every now and then there is a mistake, and that's a mutation. Now, so the replication, you can see on here, it's happening in this direction. On here, it's happening in this direction. I don't know if you read the chapter, but your book is going to refer to that replication takes place from 5' to 3'. And when it's saying 5, it's talking about the 5th carbon on the sugar molecule. If it's saying 3, then it's talking about the 3rd carbon on that molecule. So I'm not gonna ask you details on that, but I'm just gonna tell you what's happening here. So think of this as a 5' end. This is 3', and the replication is gonna happen in this direction. Here you have 5' on this side, top, and then 3' on this side. So it's gonna happen in the opposite direction. Same enzyme, DNA polymerase, is adding the nucleotides on this side and also on this side. But because this is happening in the opposite direction, this is called the lagging strand. It kind of lags. This is called the leading. So the original, this strand is leading strand, this is lagging strand. So when this is happening in the opposite direction, sometimes what happens, it leaves a little bit of breaks in between. So maybe it forgets to add one nucleotide. So what's gonna happen, another enzyme called ligase is gonna come in and it's going to fix those breaks. It's like a glue. So think of ligase as a glue. I think I wrote it down on the next slide. So enzyme called helicase is going to unwind the DNA. Then enzyme called DNA polymerase is going to add the nucleotides that are complementary to the original strand. This is the original strand. So this is the original strand, parent strand, and this is the newly built strand. And on this side, this is the newly built strand, and this is the original strand. That's called semi-conservative. Semi-conservative means it's gonna contain one parent strand and one new strand. And then because this is happening in opposite direction and this is the lagging strand, it leaves some breaks sometimes. So the enzyme called ligase, it comes in and it starts to fill those breaks. It starts to form some covalent bonds between the adjacent nucleotides. So it starts to repair that. So this is like glue. So this is the enzyme that's like glue. Now, before I move on, I want to know if you guys understand the difference between 3 enzymes and the function.
So out of curiosity, I just want to know, DNA polymerase stands for, let's say is a builder, it builds, right? While ligase is some sort of glue, when helicase unzap it, it will use ligase to kind of attach it back, right?
It uses the ligase for what? I couldn't hear the last part.
To kind of attach it back, right?
Yeah, so if there are any breaks when the DNA polymerase is adding these nucleotides, then the ligase is going to come in and kind of glue them together.
Okay, yeah.
So just to repeat it one more time, helicase is going to unzip. It's going to open this book because you want to read the information. DNA polymerase is going to grab the nucleotides in the nucleus and going to start adding the complementary nucleotides to the strand, parent strand. And then the ligase comes in and works on the lagging strand. If there are any breaks, it closes them. So it's all nice, like this one. And then in the end, you get one parent strand and then one newly constructed strand, and that's called semi-conservative. And all this, it's happening really, really quick, like a machine gun. So let me see if I can find you a video for that. So after replication, each DNA molecule has one parent strand and one daughter strand. This new strand is known as daughter strand, and that's semi-conservative. So let me show you a video before I move on. So here's one. It's happening really, really, really quickly. So let's see, maybe this one. So here you can see the double strand, and then you can see the single strand here after it's unzipped. So this is a helicase there. So that blue is helicase, it's unzipping it. One is going this way, another one is going this way. And then the other enzymes are constructing it really quickly. Here's another one. Primase is an enzyme that just basically tells the polymerase where it needs to start. So it tells that this is where you need to start building. So polymerase comes in, starts adding the nucleotides, and then and you can see it has 2 strands, and then if it's a lagging strand, ligase is going to come in and close the brakes. So here, unwound DNA, so this would be helicase here. It's unwinding this DNA here. And what I mean by semi-conservative, this is your original template. It's blue. And then this newly synthesized strand is red. So here you have one blue and one red. So one is parent strand and the other is the daughter strand. So again, same thing here. And this is what refers to semi-conservative. When they were trying to figure out how that happens, they came up with these different terms here. They said, oh, the original stays together, you just make a whole new one. And then here they said maybe it's partial, but then this is what's really happening. One old one stays, new one is built. Old one stays, new one's built. So from one strand, one double helix, you have 2 here. Then from 2, you have 4 here. And from 4, you're going to have 8 here. And nowadays, when they're doing testing, they can do this in the test tube. So they can basically take the DNA they want to copy, they can add it to a test tube, then they can add the A's and G's and T's and C's, and then anything else that's needed, maybe the enzymes, the enzymes. So they're going to add the helicase, maybe they're going to add the polymerase, and then they're going to just put it in a machine, and the machine is going to copy. So I don't know, I think in 3 hours, it can create like billions of copies of this one single strand of DNA. And they do that for research. So that's DNA replication. 3 important enzymes are DNA polymerase, helicase, and ligase. Another enzyme I asked you guys to know was RNA polymerase, and that's in transcription. It does the same thing as DNA polymerase. It reads the information and adds the nucleotides, but in that case, it's adding the uracil for RNA. So that's RNA polymerase. So there's a question for you guys. So this is the image that you have. This is the original strand. So which of following best represent the newly created DNA strand after replication is done? So this is A, this is B.